Hello, everyone. Today we are here for the next our TSS presentation with Nick Shobin and Crystal Cruz from Quantix company. And I'm going to little bit about this technique and then the introduction of our speaker. So the Quantix assay development approaches, it is very ultra sensitive. We can say like 100 and 1,000 times than traditional ELISA assays, and they have the wonderful, because this is a very sensitive, sometimes we are not getting the result because of the we are losing the signal, so maybe there is no enough concentration. So they are going to talk about this technique. And so we have a Crystal Cruz. She's a business development manager, and their background is like a cell therapy, and she did a MS in the immunology from New York University, where she started like a manufacture like dendritic cell vaccines for the phase 1 and phase 2 clinical trials in the GMP settings. And after that, like Quantrix, she worked with the military biotech for the 8 years as a technical consultant. So, and we have another speaker, Nick Shubin, and he's a field application scientist, and he's trained and worked closely with the customers on the various LC development projects. And he earned his PhD in pathobiology from Brown University in 2013, sorry. And prior to joining the Quantic in 2019, he completed his postdoc from University of Washington and also Seattle Children's Research Institute. Nick has almost contributed 40 peer-reviewed publications and focus in the immune regulation in acute inflammatory conditions, such as like a sepsis or other diseases. So please welcome Crystal and Nick.
Thank you so much, Arjun. I really appreciate the introduction. And good afternoon, everybody. Thank you very much for attending our presentation today. As Arjun said, my name is Crystal Cruz, and I am your local business development manager. I am based in New Jersey, and I'm your main point of contact at Quanterix for any sales-related questions or quoting, etc. But I can also help triage your technical questions and pass them on to our experts behind the scenes. So when in doubt, you can always reach out to me first, and I can pass along your information to the appropriate contacts. And so one of our internal experts is here with me today. Nick Shubin is your local field application scientist, as Arjun pointed out. And he and I work very closely together in Pennsylvania, Delaware, and New Jersey. Nick also is based in New Jersey, so we are both a short drive away from Jefferson. If ever you get your own instrument, Nick will be the guy to train you. So Nick is going to walk you through our technologies and show some data from homebrew applications that he and others have worked on with customers. But before he goes into the details of our tech and these custom assays, I just want to first address our vision as a company and where we think our technology is going and where we hope it can take you along with it. So without further ado, and please stop and ask questions at any point in time. I don't think there are very many people on this call, so we can keep it really informal if you like. This slide just takes you through the progression of illnesses such as cancer, neurodegenerative disease, cardiovascular disease, etc. And generally speaking, as we're all aware, as you become increasingly more sick, biomarkers become more concentrated. But these proteins are also pretty difficult to access. They usually require a biopsy or something invasive like a lumbar puncture. And in fact, for many neurological disorders, specifically like CTE, you can unfortunately only diagnose during an autopsy. And so this is what we consider sick care, when a person is often far too along in the process of the illness for any meaningful intervention. And our hope is that we can get to a place where we can detect disease before the person becomes symptomatic. That would allow time for the application of therapeutic and preventative measures. So it's Quanterix's goal as a company to help researchers do this by developing instruments and assays that are ever more sensitive. essentially allowing medicine to peek behind the veil of human health per se. And right now there are about 205 FDA approved proteins for diagnostic use, and about 1,300 proteins that are what they call lab developed tests, or LDTs. But there are an estimated 10,000 more proteins in serum that can't be measured without an ultra-sensitive platform like ours. And that's really where the cutting edge is, and especially in neuroscience and oncology. And this is where we hope our tools can help you envision new possibilities for healthcare by giving you the opportunity to see deeper into a person's health than ever before. And just for some specifics, as Arjun mentioned, the Simoa platform is roughly 900 to 1,000 times more sensitive than ELISA's. We can robustly measure down to about femtogram per mil concentrations with high precision. We often achieve CVs of about 10% or more, or lower, sorry. And even at the super low concentrations. So we're really well suited to measure these key biomarkers. And we think that protein biomarkers specifically are a key to understanding human health, since they're essentially the result of nature versus nurture, right? So how well you sleep, you know, what your diet, do you exercise. I'm sorry, okay, I didn't realize I can mute people. So. You know, how well you sleep, all these environmental factors, where you live, exercise, etc. All of these factors laying on top of your genetic background actually determines your health, right? And so we feel that protein biomarkers tell the story of your health the best. And so some of you may or may not be aware that Quanterix actually has two ultra-sensitive platforms, one that uses a bead conjugated to capture antibodies, and the other a planar system that uses plates with the capture antibody printed onto the well surface. I know in the title of the talk and in the invite, we mentioned specifically the SPX system, but after talking with Nick, we decided that we'd like to showcase both platforms today. So here you can see our HDX and SRX systems for our bead assays. The HDX is our automated platform, and the SRX is semi-automated. And then the SPX is our newest addition. Just to point out, these two instruments use our bead array system, and also just to note that most of our neuroassays, or I should say all of our neuroassays, have been developed on this platform. So when you hear Simoa, which means single molecule array, or when you hear Quanterix, often this is the platform that most people are thinking about. And then the SPX is our newest addition, and it utilizes planar technology while also being quite sensitive. And so we'll look at some custom assay development on both of these platforms. So the last point, we do offer contract research services at our accelerator lab based in our headquarters in Billerica, Massachusetts. We offer simple and straightforward sample testing, but we also offer assay development services as well. So if there's an analyte that you'd like to measure, but we don't have a commercially available assay for it yet, we're happy to develop the assay for you and either run your samples, or we can transfer the assay back to you for you to run on your own instrument if you have one. And if this is something that you're interested in pursuing, feel free to reach out to me and we can discuss in further detail. And just on a side note, Arjun asked me if there are any instruments that are available locally. And I wanted to bring to your attention the lovely group at the Human Immunology Core at UPenn. They also, they have our HD1 system that you can see here. They also offer sample testing and homebrew assay services as well. So you can, feel free to either Google them, or you can reach out to me, and I'm happy to put you in contact with their core director, Ling Zhao. And so that's it for the introduction. Does anybody have any questions about any of what I mentioned right now? Okay, so I'm going to unshare my screen and I'll pass it over to Nick, who's going to take you through the principles of our technology, as well as these custom assays.
Can you see my screen?
Yep.
Okay, great. So yeah, so I'll just go ahead and start with just kind of the background of the technology for our instruments. So I'm just going to start with our bead-based technology. So our bead array technology uses two instruments, the HDX, which is our fully automated instrument, and also our SRX, which is our benchtop version. And so both of these instruments use the same technology, which I'll talk a little bit more in the following slides, but they both use what we call Simoa disks. And so on each of these disks are small arrays here. And so I'll again talk more in detail. But both of our instruments, or all of our instruments, are capable of getting down to this femtogram per milliliter level of detection, and which wasn't really no instruments were really capable of doing that until about 2014, when our instruments kind of came into the field. So, so just a little bit of basic background on how the bead arrays work. They're really just your basic bead based sandwich ELISA. And so kind of how it works is you have a paramagnetic bead that has capture antibody attached to those. And as you run those through the instrument, you'll add a sample will be added, which has the antigen that you're looking for in your assay. And then the next step would be to add biotinylate detector antibody. And then the last step is to add your streptavidin conjugated to an enzyme. And then the final step is a substrate is added. And if you have a full immune complex attached to your to the beads, then that substrate is cleaved into a fluorescent molecule called resirufin. And so essentially what happens with the bead-based system is around 15 to 25,000 of these beads are loaded into a Simoa disk here. And I have more like workflow type stuff, kind of slides coming up so you get an idea of those. But basically those beads are loaded in here. And then in these small spots here are arrays, and those arrays include 239,000 femtoliter sized wells. They're just big enough to hold a single paramagnetic bead. So you can see what those kind of look like here in like an electron microscope type picture. And so, so that's sort of like the basics of the technology. Again, it's like just basic bead ELISA, but using this kind of platform. So kind of getting into, why is this so sensitive? And so this is kind of a picture, just like a graphic kind of describing what happens inside of those wells. So once the beads are loaded into these arrays, this is like a depiction of each of those arrays. And so if you have a case where you have more beads in your assay than there is analyte in your sample, you'll get this sort of on versus off signal. And that's really how we get to the super sensitive levels in our assays. So we call this digital ELISA, so this on versus off signal. And then there's also cases where you may have more analyte than you have beads in your assay. And we call this analog ELISA. So this, what we can do is actually, instruments can actually measure up to 16 to 20 immune complexes per bead. And so when we put both the digital ELISA and analog ELISA together, the instruments can do more than 4 logs of sensitivity starting at that femtogram per milliliter level of detection. And so, yeah, please stop me if there's any questions along the way. Certainly happy to answer any questions. So just to kind of give you an idea of why this is so much more sensitive than just traditional ELISA. So with traditional ELISA, usually the reaction volume is like 100 to 200 microliters per well. And because of that larger volume, there's a lot of diffusion that occurs in the traditional ELISA, and it takes millions of molecules to reach that level of detection, the detection limit. But with Simoa assays, the reaction volume is 50 femtoliters, which is 2 billion times smaller than what you would have in traditional ELISA. And also there's oil that seals in that signal as well in the assays. And because of that, diffusion is really defeated, and we can get to single molecule resolution. And that's how we get to like this ultimate sensitivity for the assays. So this is just an idea of the assay workflow, and just kind of give you an idea of what happens with the HDX, which is our fully automated instrument. So essentially what you do is you load in your detector, bead, sample, diluent, all of the reagents just go into the instrument, and then you tell the instrument where your samples are on your plate, and you just more or less press start. And once you do that, what happens is the beads are mixed with your sample, and then they're incubated for a certain amount of time and then washed. And at that point, you'll have your analyte of interest on the capture antibody, which is on the beads. And then what happens is detector antibody is added in the next step, incubated, washed. Enzyme is added in the next step, incubated and washed. And so at that point, you have a full immune complex attached to the beads. So what happens then is the beads are actually mixed with the substrate, which you can kind of see here. So the beads are kind of loaded into one of the arrays here, and this is kind of like a sideways view of what the arrays would look like. And so the beads come into the arrays, and then there's a magnet that kind of helps pull the beads into the arrays here. And then oil is sealed, seals that signal in. And then what happens is if there's an instance where a bead has an immune complex attached, it will cleave the substrate so that there's a fluorescent signal. And then there's cases where there's no immune complex attached, so there's no fluorescent signal. And then there's many cases where there's just no beads in general. So then what happens is there's a series of images that are taken of the arrays. And then the instrument will just provide you with a calibration curve and provide you with all the concentrations for your samples from that curve. So just kind of getting into some applications, and we know that you might be more interested in sort of like your own assay development approach. So we kind of focused on those. And so the way that we do our assay development approach, we call them homebrew assays. And it's really the same. The assays run exactly the same way as they would with any assays that we sell. But what you do is you will conjugate your own antibodies to magnetic beads. So you would take your own capture antibody, conjugate those to beads, and also biotinylate your own detector antibody. So usually what works best is if you already know you have an ELISA that works pretty well already. So say you're getting into the picogram per milliliter level of detection, then you can apply those same antibodies to the Simoa technology and typically get 100 to 1,000-fold more sensitive results. So just kind of getting a little bit more detail about how the homebrew process works. And so if you're ever interested in this, then I can come and train you on this process as well. So basically what you do is you would have your detector antibody in this case, and down here is for capture antibodies. So with your detector antibody, you'll just buffer exchange it into a biotinylation buffer, and then just add in biotin. And we usually suggest a 40x ratio to start with. So you'll have 40 times more biotin than you would have your capture or detector antibody. And at that point, you'll buffer exchange to remove any extra biotin in the, and then you'll be left with your biotinylated detector antibody. And so for the capture antibody, what we do is we buffer exchange it into a conjugation buffer, and then we will EDC activate the beads. So EDC is a chemical which just basically makes it so that the beads are receptive to antibodies, more or less like sticking to the beads. And then we add a blocking reagent just to make sure there's no non-specific binding to the beads. And then there's a final cleanup step. And then you're left with your beads that have a capture antibody attached to them. And then also with our homebrew starter kits, we offer an antibody set that we know works really well with these assays. So it's a prostate serum antigen assay or antibodies. So you can basically just run these antibodies in parallel with your own antibodies just to make sure that everything's kind of going the way it should. So we have a couple examples of some homebrew assays that have been very successful, and both of these were published just this last year. So the first example is with blood phosphorylated tau 181 as a biomarker for Alzheimer's disease. And so just some background from this paper is that there's more than 50 million people worldwide that have dementia. And they note in the paper that there are other markers that can accurately detect Alzheimer's disease pathology, but they're both, either invasive, so you have to get, cerebral spinal fluid from people to look at their amyloid beta or tau levels, or they're very expensive, like for instance, PET scans. And so I recently heard that, that this can cost like around $5,000 per person for a PET scan. So, so they wanted to try and find a biomarker that may be cheaper and more accessible to use. So they had found that CSF tau phosphorylated at 39181, so p-tau 181, is a highly specific biomarker for Alzheimer's disease pathology. So they decided to try and apply that same approach to blood, and to see, with the hypothesis being that p-tau 181 from the blood could accurately discriminate between cognitively unimpaired people from Alzheimer's disease patients. So the way that they did this is they took a capture antibody that they knew was specific for p-tau 181 and conjugated that to beads. And then they bioattenuated just a total tau antibody as a detector antibody. And then they used this to detect p-tau 181 in the plasma. So what they found was they actually used this with 2 different cohorts. And in the first cohort, which included 226 people, they found that plasma p-tau 181, actually, those levels increased as Alzheimer's disease pathology increased as well. So you could see that in people that were young individuals, people that had mild cognitive impairment, all the way to Alzheimer's disease, those levels kind of increased as people, continued towards Alzheimer's disease. And then in a second cohort, they actually found the same results with Alzheimer's pathology. But what was really neat was that they also looked into people that had different types of neurological diseases, like Parkinson's disease, and found that they actually did not have increased levels of p-tau 181. So this looked like it was looks like a marker that can not only discriminate, Alzheimer's disease as it climbs along the continuum, but also can discriminate between different types of neurological disease as well. So another example of a homebrew assay that I think is really neat is this is a 12-plex homebrew assay. So this is a pretty complex homebrew assay approach. But what they did was they essentially conjugated different types of the coronavirus protein to capture beads. So they used the full spike protein, the S1 protein, receptor binding domain, and the N protein from the SARS coronavirus 2. And basically, the capture beads that they used have different fluorescent probes attached to them so that they could discriminate between those types of proteins. So the instrument can actually measure those different probes as it runs through. So that's kind of how the multiplexing assays work for on the HDX and the SRX. And so what they did with their detector antibodies is they actually used 3 different plasma samples. And they used anti-IgG, anti-IgA, and anti-IgM as detector antibodies. So in the end, what they were able to do was to find host antibodies that could recognize the different coronavirus antigens. And then also they were able to determine what type of antibodies those were by using these different detector antibodies. So this is just one of the major findings from that paper. So you can see all the different types of data that they got, with the different antigens that were measured and also the different antibodies that they looked at. And what they saw, they looked at, people pre-pandemic, so they kind of had a baseline for antibodies in these people. And then they looked at PCR negative patients. So they actually found a number of false negative patient samples, as you can see here. And then you can see as people were infected and as time went on, you could see that their antibody levels also increased. So this gave them, a pretty high fidelity look at how, serology is working in people that were infected with coronavirus. So are there any questions on kind of the bead-based approach, like the assay development approach? Okay, so I'll just briefly discuss our commercial assays. And so we offer a number of commercial assay kits. And so those come as either what we call advantage kits. We have a number of multiplexing assays and also discovery assays. So for our advantage assays, these are a little bit more validated in-house. So we'll run those on multiple instruments and also across multiple lots, just so we know that these assays are very consistent from lot to lot. And then with our discovery assays, these tend to be newer assays that will probably eventually become advantage assays. They just haven't quite gotten to that level of validation yet. So So these are both, extremely sensitive assays, but just to give you an idea of kind of what we have to offer. And so it also, as Crystal pointed out, all of our neurological based assays are on our Bead platform, so you can get an idea of what we offer for those. So this just gives you an idea. If you were ever interested in one of our assays, you could come to our website and take a look at like our data sheets. And so this is an example with neurofilament light, which is our most popular assay. So it's thought to be sort of a broad neurological marker. And so what we show in our data sheets is typically healthy levels from in different matrices. So we have like plasma serum, CSF, and mouse serum and plasma, just so you can get an idea of what you would expect with our assays. And then we'll show you kind of the mean average levels you would get in healthy individuals and the level of detection, how many of those healthy individual samples were above the limit of detection. So if there's any questions on the Bead-based platform, I'll kind of move into the planar array technology. So our planar array technology, and I'll kind of go in more detail about how it works with these assays, but really the benefit of our planar array technology is that we can do increased plexing. So we can do up to 10 plex assays with our planar array technology. And also these assays tend to be more focused on like immunology, inflammation, oncology type assays. But these our planar array technology is also capable of getting down to femtogram per milliliter level of detection. So we can get super sensitive results with all of our platforms. So sort of the way that the planar array technology works is it's really just your basic sandwich ELISA format, but we essentially spot capture antibody into the circular pattern onto the plates. So so you would get a plate that looks like this, and if you this is an example with our human core-plex cytokine assay. So it's our 10-plex assay, which is our highest plexed assay. And so what you can see is that the capture antibody is spotted in like a circular pattern here. And what you can see is, so each of the spots represents a different capture antibody. So you have like IL-12 P70 in one spot, IL-1 beta in another spot, and so on in this pattern. So if you kind of look at the plate in a sideways format, so if you looked at like IL-1 beta, this would be your IL-1 beta capture antibody here. And then as you run the plate through the instrument, you would, just like a normal sandwich ELISA, you would add your sample, which contains the antigen. So it could be like IL-1 beta in plasma. And then you would, essentially incubate that, wash off any excess, sample, add in your detector antibody, which is biotinylated. Then the next step is to add in an enzyme, which is streptavidin conjugated to HRP. And then the final step is to add in a chemiluminescent substrate. So this is cleaved by the enzyme if there's a full immune complex attached to the capture antibodies. And at that point, we'll take the plate and we'll put it onto the imager, which I'll show you in a minute. So another thing that makes these assays so sensitive is that we've optimized the mixing reaction. So again, you can see that the format is in this circular pattern. So when we put the plates on our shaker, essentially what happens is you have the volume of liquid kind of tends to look like this. So this causes the antigen to kind of be driven into the capture antibodies here. And so we also had a paper that was published on this showing that they actually tried to put antibodies in the middle here, but there's a sort of a depleted region. So that's sort of why we have that circular pattern. So then the final way that these assays are so sensitive is that we use a high precision CCD camera, and that just leads to very low background signaling. So what the instrument does is once you've taken it through the incubation steps and the washing steps, you put it on the imager. And so what the first thing is it does is it takes a relatively long exposure. So it's about 5 minutes for the first exposure. And so if you have your calibration curve here, so say you have your high concentration calibration points here down to your low calibration points here, the long exposure will allow for the more like the lower end of your curve. So that's how we get to the femtogram per milliliter measurements. And then there's a much shorter exposure. So this is how we get to the higher end of the curve, essentially. And so what we do then is the image files are taken to the software, which is you will just load it onto your own computer. And what it does is it takes those two images, and there's an algorithm within the software that can basically put those two curves together. And then you are able to get 4 logs of sensitivity, starting with femtogram per milliliter level detection. So this is just going through the assay workflow. So this is an example if you have, say, you're looking at a 3-plex assay. So when you get the plates, they actually have a blocking reagent that is coating them. So the first thing you'll do is you'll take your plate over to the microplate washer. So I guess I should back up. These are the 3 components that come with the SPX. So we have a microplate washer, a microplate shaker, a microplate washer, and then the imager here. So you'll take your plate to the washer, wash off the blocking reagent, go over to the bench, and then aliquot on your samples, take it over to the shaker. And so at that point, you'll see that you'll start getting antigen attached to the capture antibodies here. So after those have shaken and incubated, you'll take it to the washer, wash off any extra sample, come back over to the bench, add in your detector antibody, and then you get detector antibody attaching to the the immune complex. So you do all of those steps to the point where you'll add in your substrate, and then you'll take the plate over to the imager here, and then it's imaged, like I mentioned in the previous slide. So we also have a homebrew assay development approach for the SPX as well. And so the way that this works is what we do is we actually conjugate on a peptide to the capture antibody here. And so when you get your plates, they actually have an antibody that's spotted to the plate here that recognizes that tag peptide. And then the other thing you'll do when you develop your own assay, similar to the Bead-based approach, is you'll biotinylate your own detector antibody as well. So so there's basically just like an extra step here where you'll add in your capture antibody in the first step and then run it through the workflow like I mentioned in the last slide. So we currently offer just a single-plexed assay for our homebrew approach, but we're working on developing and releasing a multiplexed approach for our SPX as well. Was there a question that came in? Okay. So just to give you an idea of sort of the next thing we do when we do like a homebrew training, or even if you're just going to develop your own assay, is to do a screening approach. So so these assays with the SPX assays actually contain IL-6 antibodies that we use as control antibodies, just to make sure that the process is working. So in the first part of your plate, you would just measure IL-6 that comes with the kit. And then what we can do is you can actually test up to 8 pairs at first, just to kind of get an idea of what might work best for your assay. So this just gives you an idea of the approach you could use if you wanted to screen multiple antibody pairs for whatever you want to look for. So we have just a couple examples of the homebrew approach for the SPX. So a colleague of ours actually did this in our demo lab at Quanterix in near Boston. And basically what he did was he made an assay to P24, which is a capsid protein for HIV. And so he took a capture antibody and conjugated the anchor peptide to that capture antibody, and also biotinolated his detector antibody, and then ran it through the assay. And so this is the sort of the typical data that you would get for your calibration curve for the SPX. So the gray points are the calibration points on the curve, and then the green points are where your samples are. So just to give you an idea of what that would look like. And so when he did this approach, he was able to get a limit of detection of 1.1 femtogram per milliliter, a limit of quantification of 14 femtograms per milliliter, and also did some spike and recovery, and was able to get a nice recovery with the assay. And then just to kind of point out like the sample volumes that you would use. So the SPX uses 50 microliters per well. Typically, the minimum required dilution for these assays is like 2 to 4 fold. So for each test, for each replicate, you would use probably 12.5 to 25 microliters of, serum or plasma or whatever matrix that you're interested in. And so then this is actually from a customer that I did training with. And so they let us share this data with you. So we're not able to share the analyte itself, but just kind of give you an idea of, what you might be able to achieve with your own homebrew approach. So with the pair that they used, and they had tested this pair already with ELISA, so they had, they knew that it worked really well before they applied it to the SPX homebrew approach. And what they were able to see was an upper limit of quantification of 50 picograms per milliliter down to an LOQ of 12.2 femtograms per milliliter. And then they actually achieved a limit of detection of 99.4 attograms per milliliter. So this is also kind of the data that you would see from, the output data. So what you see here are the calibration points that they used are on this side. And then these are the intensity values. So the raw data that you would get from the instrument is here. And just to kind of give you an idea of like the CB values that they saw. And so this was just really like the first pass assay that they were trying out. And then recently they reached out to me to tell us that they were seeing down to 0.2 femtograms per milliliter range for this assay once they had optimized it a bit more. So just to kind of give you an idea of what of a different customer, what they were able to achieve. So in terms of our planar array commercial available kits, this is the menu that we offer for our human assays. So already kind of went through the cytokine panel. So this is the 10-plex assay that we offer. We have a few other multiplex assays. We have a chemokine panel, angiogenesis panel, and also another biomarker panel. And then we have a number of single plex assays. So these are what we call our developer assays. So these just haven't been quite as validated as our core plex assays, but are also quite sensitive. And so if you were going to look at our website and we're curious about what you might expect with our core plex assay or cytokine assay, again, we in-house will test around 20 or so healthy individual samples, and then this is kind of what you would see. This is what you would see for our core plex assay. A limit of detection in this range for these cytokines, LOQ here, and then we'll share the LOQ and limit of detection for those analytes as well. And so this is just a slide kind of showing, the differences between the three systems that we offer. So over here are bead-based platforms, and then our Simoa planar array platforms. And so just if you were curious about, how many samples you could use per plate, how long it takes to run those, how much hands-on time is involved in those types of questions. So that's pretty much all I have to share today, but certainly happy to take any questions or any comments. And so thank you very much.
Thank you, Nick and Castro. Session open for question and answer. If you have asked any question, please ask.
I do have a question. May I ask?
Sure.
Sure.
It was a really interesting talk. So I have two questions. First one is, since this is very sensitive, what's the limitation for this ELISA? And then my other question is, can we diagnose like genetic disease related sickness with this ELISA? That's all.
Yeah, I'd say that anything that you're trying to do with ELISA, you can take it to the Simoa platform. Usually, it's really best if you already know that your ELISA works, and then if you take those antibodies, you can typically get 1,000 up to 1,000-fold more sensitive. So if you're using ELISAs for the application you mentioned, you certainly could use it for in this platform as well.
And what's the limitations for this ELISA, like digital ELISA?
So for limitations, I would say I think that if you're interested in sensitivity, if you want more sensitive results, then this is the way to go. I think if you're people tend to use ELISA if you already have, a range of, biomarker detection or protein detection that you're happy with, then, I think then ELISA is the way to go. So say you don't need sensitive results, then, ELISA, traditional ELISA may be better. But one thing that is nice about, especially like our HDX, is it's fully automated. So even if you have, an ELISA, but you don't have, many hands to help, process ELISAs, then you can, transition it to, an HDX and basically just put in all of your reagents and have an automated workflow.
Thank you so much.
Any other questions, guys?
I don't think so.
Okay.
Nick, I have one question.
Sure.
There's one thing I have noticed, I'm working with the BDNF, right? So I'm using like a normal, traditional one, because I have samples from like a tissue perfusates, because I'm using the same tissue from like a rat. So I'm using same tissue for like organ bath studies. And during those studies, I'm taking those tissues perfusate, right? So the traditional one is not very sensitive, but I have to go and see if it is working, because I'm using tissue as well as like a cell culture perfusate. So what do you suggest if my picogram BDNF is not like much more in their tissues, so how we can increase the sensitivity if I have this issue?
Yeah, so people certainly, use different matrices like tissue culture sample, and cell culture supernatants. So a lot of times an issue can be that there's matrix related effects. So if you're diluting say fourfold and you're not getting good your spike recovery results, with that same tissue matrix. One nice thing is that you might be able to dilute that much further and run it on an HDX or an SPX and then you're kind of getting away from those matrix related issues. So yeah.
Thank you so much. Yeah. So we don't have any questions yet.
Okay. Well, you all have our contact information now. So if you have any questions and you want to take it offline, feel free to reach out to Nick and myself, and we're happy to help.
Thank you so much, Nick and Christo.
You're very welcome.
Thank you.
Thank you for helping organize this. It was great. All right. Bye, everyone. Have a great weekend.
You too. Thanks.
Bye.